to Yocha Dihi Winton Nation for sponsoring today's video. Hello, everybody, and thank you for joining us this week for our Lunch with a Scientist. Today, I'm very excited to have Dr. Anya Brown joining us. She is a marine scientist and assistant professor at UC Davis, based in Bodega Marine Lab in Bodega Bay, California. She studies marine ecology and takes particular interest in the microbes that are associated with hosts such as corals. Her work has taken her to the French Polynesia, the Cayman Islands, Puerto Rico, and St. Croix, as well as Massachusetts. So a bunch of really amazing places. She's originally from New York, but has moved around a lot of places in order to gain some experience as well as go to graduate school. So with that, I'm going to go ahead and bring in Anya. So hi, Anya. Thank you so much for joining us for Lunch with a Scientist.
Hi, my pleasure. I'm excited to be here.
I think that the kids are really going to enjoy what you're talking about today because anytime we can bring in gross science to a classroom and they'll see what that means in a little bit, it always is a big hit. So I just appreciate you sharing your work today. We do like to have all of our scientists start a little bit by talking about how they got to their career. And then, so if you want to go ahead with that, I'll maybe ask you a couple of follow-up questions or interject here and there, but go ahead and give us a little bit of background about how you became a scientist and the work that you do.
Great. I'm so happy to be here and tell you a little bit about my journey as a scientist. So I really like this picture too. So the background of it is supposed to be microbes that a friend of mine who's an artist drew based on one of the microscope slides that I had sent, pictures from a microscope slide that I sent her. And so I'm a marine biologist, but I'm also an ecologist. So a marine biologist, I study organisms that live in the ocean. As an ecologist, I study the way organisms interact with the environment and with each other. And I also am a microbial ecologist. So I study the teeny tiny microorganisms present in the marine world. And maybe you don't often think about the microbes because you can't see them, but microbes are everywhere. You have microbes on your skin, you have microbes in your gut to help you digest food. There's microbes or bacteria in the roots of trees, helping them gain nutrients so they can grow. Pretty much every living organism has some types of microbial association. So I like to use this little figure here where it shows a host. So like humans, we're a host to a bunch of different microbes and this cloud of microbes around them to really show that we're more than just our tissues. And the animal that I like to study the most has been corals, but I've never, and the bacteria and archaea and other microbes, these teeny tiny organisms that live within them and on top of them in marine environments. But I haven't always wanted to study microbes. In fact, I never really thought about microbes at all when I was growing up. And so I grew up in New York. And that's where a lot of my family lived. And so I have here just a little picture of some of my family members who live in the New York area. But my mom is originally from Maine, so I would spend a whole lot of time in Maine as well. And so it was on the rocky coasts of Maine that I started getting interested in marine biology and marine ecology. And so it was probably somewhere around middle school or elementary school that I got really into it, in part because I read this book by Madeleine L'Engle called A Ring of Endless Light. And I don't know if that's still a popular author. She wrote A Wrinkle in Time. But in this book, she could, one of the main characters could communicate with dolphins. And I thought that might be pretty cool if that were possible. And so I started thinking more and more about, well, how much fun would it be to at least be a marine biologist and be around the ocean all the time? And so to start to get some of the feel for what it was like to do research or think about the things going on underwater, I attended a camp because that's sometimes what nerdy kids do. And this one was called the Acadia Institute of Oceanography. And it was based in Maine. And so I got to explore the rocky intertidal zones, explore the sandy beaches all around the coast of an island in Maine that I had spent a bunch of my summers growing up going to with my mom and my grandparents. And then when I hit high school, I realized, like you could do this research thing. And I started learning more about the process of becoming a scientist. in a class called Authentic Science Research. And it was a pretty cool and an intensive experience. And maybe some of you have done something like it where I picked a topic. In this case, I wanted to learn about the antibacterial activities of seaweed, which is the one that's right here. It's a seaweed called nori or porphyra, and it's the same seaweed that you see wrapped around sushi when you go to a Japanese restaurant. So I was interested in the antibacterial activities associated with the seaweed. I did a lot of literature searching for my first year in the course. My second year, I found a mentor at the University of Connecticut and did some experiments. And then in the third year, I presented it to different science competitions around New York, where I grew up, and entered it into the Intel program, which I think at the time was the Intel Science Talent Search, where I was a semifinalist. And so I was like, oh, this science thing, maybe there's something to it.
Do you think like having a mentor at a young age was beneficial for you?
Oh, yeah. I mean, I think I gained a ton of confidence in my skills as a scientist, both to be able to communicate with a mentor or with someone who has a PhD, or think about what research really is. And the other part of this that was really important was I became familiar with how to read a scientific paper. And a lot of people don't get exposed to that until they're in college, if they ever get exposed to it. And so there were just some really good foundational skills that I learned that I didn't even realize how important they were until I got to college and beyond.
Do you think that helped you progress in your career entering as a professor, knowing like, I got mentorship from someone else, like I want to give this back as well? Like, do you think that helped with that?
That was maybe like the first, I think all throughout school, you get really good mentors, and going into this field, you're going to be a mentor in a lot of different ways. And so that was one part of my like pre-college experience that solidified like this need for having some type of mentor. And my next experiences with other mentors solidified that even more. And it helped me realize the characteristics of a good mentor.
Well, thank you so much. I'm going to let you go ahead and continue. I just had a couple of follow-up questions because the international, I love seeing students that went through the science research process. Now in your position, it's very inspiring for our kids that are doing this kind of work. And you see the results. You know, we have a professor here that's doing amazing research. So I'm going to go ahead and let you continue, but I just wanted to interject with that. So thank you.
But after gaining all of these experiences, I wanted to go to college, and I wanted to go to college and continue learning more about marine science and marine biology. But I also really wanted to be challenged. So I looked for schools where I would be both challenged and I could major in marine biology. And I decided to apply to Brown, and I got in, which was awesome because at the time it was one of the few Ivy Leagues that had an option to be a marine biology major. And so while I was at Brown, my interest in ecology is what it started. I got the ecology bug while I was at Brown. And that was in part because I was involved in this freshman year seminar, moved across the country to California, where I attended California State University in Northridge for my master's. And part of that experience was getting involved with the Moorea Coral Reef LTER. So that's back in French Polynesia. And they have a long-term monitoring project where they're learning about all of these different aspects of the ecosystem. And so it was through spending some time in Moorea and reading a bunch of papers, just like what I was doing when I was in high school, I learned that I was really interested and excited about corals like this one here, and algae like this fuzzy stuff on the rock here, and how they compete with one another. Because when they're touching like this, you can see they're like actively competing, where the coral in this case is starting to overgrow some of the algae, or the algae will start to overgrow some of the coral. And in fact, usually it's the algae that outcompete or negatively affect corals. That's what this arrow means. And in the process of looking into coral algal competition, I started getting interested in microbes because, as it turns out, microbes play a role in the way algae interact with corals. So algae produce photosynthesis, just like plants do on land. And photosynthesis is full of carbon, which is kind of like a tasty snack for microbes. So when microbes are when algae are present, it increases the number of microbes that are present too. And when you have an increase in the microbes that were present, they start to breathe a lot. They draw down oxygen and they create this mini hypoxic or low oxygen area and basically suffocate the coral, which sounds terrible, but I thought was super interesting. So that's what I studied during my master's. And again, a lot of my experiences build on each other. And so while I was in my master's and I was studying this potential suffocating of corals by algae with an interaction with microbes, I met my PhD advisor, who was interested in this process and interested in thinking about other organisms that might interact similar to corals and seaweeds. Although I was still working in French Polynesia, my home base became the University of Florida. That's where I started my PhD. And then my PhD advisor decided to apply and got a position at the University of Georgia at the Odom School of Ecology, which is where I moved as well. So I spent 6 years altogether between University of Florida and the University of Georgia, but all doing my research in Moorea and French Polynesia. And while I was at UF, I joined the Women in Science and Engineering group. And while I was at Georgia, I helped to start a Women in Science group. I was interested in these corals and algae and how they compete, and my PhD advisor wanted to learn a little bit more about how another organism might influence that interaction. And that other organism was a vermetid gastropod, Ceras maximum. The weirdest snails you've ever seen. I mean, more like a worm than a snail. In fact, they're often called worm snails. And so I'm pointing to a couple with these arrows. So there's 1, 2, 3, 4, 5 of these snails that I'm pointing to, and they're really bizarre. So when they're babies, they're floating in the water column, and they land on a piece of dead coral. So like this turfy dead part here. And once they land, they settle. So what that means is they start to meld themselves to the reef surface, to that dead part of the reef. And the reason why they go to the dead part of the reef is because the live coral itself kills them. It either eats them or it stings them and then they die. The snail sticks itself to the reef and then it remains there forever. This means it's sessile. So it's stationary. It doesn't move once it gets there. And what's crazy about them is that they have to have some type of adaptation now to eat. So what they've done is create this mucus net, which is basically just like a slime net that they put over the reef. It kind of looks like a spider web. But the way that I like to think of it is their snot. And so they collect everything that falls down from the water column onto this super snot, and then they suck in this mucus net and consume it in all of its contents. And a friend of mine likes to describe this as blowing its nose and then eating its own snot. So that's a nice thought to leave you guys with here. So I was interested in how that mess, that net influenced corals, how they interacted with algae, and how it looks like they're really trapping conditions where corals and algae are competing, and how that might negatively affect corals and help algae outcompete corals even more. And through this project, I also started getting interested on what corals you find these snails, because sometimes there are corals that have these snails and sometimes corals that don't have these snails. So with this, you can maybe start to see how there's this little bit of oh, I noticed something cool. Let me go research that. Oh, I noticed this other thing cool. Let me go research that aspect to the way that I do science. So I finished my PhD and I was really excited to continue studying corals and microbes. And so I did 2 different postdocs. And so a postdoc is after you finish your terminal degree. So in this case, my PhD, it's like an additional training period. But you're no longer a student. You're just not a professor quite yet, right yet. You're just doing research. just doing research. You're doing the best part. So I did two. One was at the University of Florida. So I went back to Florida, and there, instead of going to French Polynesia, I went to the Caribbean, to an island, or to a set of islands called the Cayman Islands. And I focused my work in Little Cayman, where the Central Caribbean Marine Institute is, and it's also called CCMI. And so part of what they had there was a coral nursery. And so my research role was to understand different aspects of the corals in that coral nursery to help inform the management of the nursery and the outplanting of the corals. And one thing that happened while I was there was a coral disease. And so a big part of my time while I was there was understanding the dynamics of the disease, both across the nursery, so across that population, as well as the microbes, so the bacteria on the corals that might be creating the disease. After spending time in the Caribbean, I decided to move, and Florida, I decided to go back to the Northeast, where I did a postdoc at Woods Hole, the Woods Hole Oceanographic Institution. And in this case, I started to work with a temperate coral that I'm looking at here. And they're pretty cool. They undergo like dormancy, which is kind of like hibernating in the winter. And so I studied the bacteria associated with the corals as they move from going into dormancy, that dormant stage, and then coming out of the dormant stage. And now I am a professor at UC Davis, which is in California. I'm based at the Bodega Marine Lab. So Davis is in the middle of the state, but I'm based at their marine lab, which is on the coast, north of Point Reyes and north of San Francisco. So I'm going to tell you guys a story about a snotty snail and some coral and their secrets. And I've been talking about corals, and I think you can probably guess what the snotty snail that I'm talking about is. But I'm going to just back up a second and talk about, well, what is a coral? And this is a picture of a mounding coral. So corals come in different shapes. This one is one that folds or creates these large boulders. And if you were to cut the whole coral in half, it would look something like this. So you would have a thin layer where the coral tissue is. So that's the part that's alive and it's growing. But underneath it, and actually the bulk of that large boulder, is the skeleton. And so the tissue is creating, or it's depositing, the skeleton and helping the coral grow taller and wider. And then the skeleton is left over. So if the coral were to die, that tissue layer would be gone, but you would still be left with that giant skeleton. And I think that's pretty amazing. So some of the corals in the Great Barrier Reef are gigantic, and that means they're thousands of years old. So thousands of years of skeleton being deposited by just this thin tissue layer that's on top. And this is also like one of the reasons why when you see reports of coral bleaching or coral death in the news or from other scientists, it's really heartbreaking. It's because it's years, hundreds of years of growth and life for the coral disappears really quickly. And on that sad note, let's talk about more parts of the coral. So a coral like this one is made up of multiple polyps. So these are interconnected units that make up the entire coral colony. So an individual polyp is connected to another individual polyp, and the coral colony itself is made up of all of these little interconnected, basically mouths with stomachs. If we were to zoom in on one of these tips, it looks like this. And if you've seen a sea anemone before, it kind of looks like a sea anemone encased in skeleton. And then if we were to take an even closer look, you could see within the tissues of a polyp, or within the tissues of this in general, are the zooxanthellae. So this is the microalgae, so microscopic algae inside the tissues of the coral. It looks something like this under a microscope. They look like little golden circles. And they photosynthesize. So like plants on land, they take in carbon dioxide and they produce oxygen and sugar. This sugar is taken from the algae, the microalgae in the coral tissues, and transferred to the coral animal. This is one of the classic and beautiful examples of symbiosis, and I think one of the most beautiful examples of symbiosis in the world, and a really important part of a tropical coral, because a lot of the food or a lot of the nutrition it gets is from the zooxanthellae in their tissues. But the zooxanthellae are not the only microbe that corals associate with. They also have a rich bacterial microbial community in their surface. So they have mucus on their surface layer. So kind of like a frog is slimy. If you were to touch a coral, don't touch a coral. But if you touched a coral, it would be kind of slimy. That's its mucus layer. And it's kind of like a protective coating on the surface of the coral that's filled with bacteria and archaea, these tiny, tiny microorganisms. And one of the roles that they play is prevention of pathogens from invading the rest of the coral. And there's also bacteria in the tissue and in the skeleton that plays the roles in defense, again, against incoming pathogens, but also plays a role in nutrient cycling, so gaining nutrition for the coral in addition to the zooxanthellae. And I think corals, one of the reasons why I think corals are so cool is because they show just tons of variation. And by variation, I mean sometimes they're big, sometimes they're small, sometimes they're branching, sometimes they're mounding. Within a coral species, you can see differences in their growth, in their size, in their color. And some of that is because there are differences then in the microalgae, those zooxanthellae in their tissues. Sometimes they have high density, sometimes they have low. As well as the bacterial communities that can change depending on the environment that the coral's in. Sometimes it can stress corals out, so have negative effects on corals when their microbiomes change, but sometimes it might be helpful for corals and help them adapt to new conditions. Big question within this is, like, when corals are changing their characteristics, like their morphology or growth rate, or their microbes, like their zooxanthellae or bacteria and archaea, is it helping corals grow? Is it helping them survive? And whatever environment they're in? And that's a big question that I'm going to talk about today, is what factors influence coral growth and their survival. Because it's really corals that are the basis of tropical ecosystems. And so in this photo, we can see all of these are different species of corals that are maintaining the life and the biodiversity of this tropical coral reef, as well as all tropical coral reefs when they're present. But corals and coral reefs are really facing a lot of stressors that are damaging them, like disease, which kills corals, overfishing, and nutrient pollution that support the loss of corals in different ways and promote the life of other types of organisms like seaweed that don't have the same characteristics that allow for a biodiverse environment. Climate change, and one facet of that is coral bleaching, which also kills coral. And so what happens when we have a reduction in coral is that these reefs go from coral dominated, so full of the biodiversity that we expect on coral reefs, to algal dominated. When you can see, it looks kind of scuzzy and slimy and gross. And so these are all different stressors, so things that negatively impact corals and lead to a shift in the dominant members of the ecosystem, reducing the foundation species, so the corals on which everything else on the reef relies. And another stressor, so a part of the reef that negatively affects corals, are these worm snails that I mentioned to you all earlier. They have mucus nets. That mucus net that cover corals is what's implicated in the negative effects these worm snails have on corals and coral reefs. They're associated with lower coral cover, so they're killing coral by making them smaller. They reduce coral growth, so we see less of that depositing of the skeleton. They lower coral survival. They lower coral photosynthetic yield, so they are affecting the photosynthesis of the microalgae, the zooxanthellae, in the coral tissues. And they lower water flow at coral surfaces. And so what this does is it effectively, like the nets are trapping water at the coral surface. And if the coral is like breathing out, so if it's producing a lot of, or it's consuming a lot of oxygen, producing a lot of carbon dioxide, it's pretty much getting suffocated when the nets are covering the coral. And it can lead to a different morphology of the coral, one that's a bit, one that's flatter compared to this more rugose, bumpy coral head. And it also has community impacts because it decreases herbivory. So it decreases the munching of fish on the seaweeds that would compete with corals. The question that I've asked in my research is, do all corals react to stressors in the same way? And if they've been exposed, if they've been in the presence of a stressor before, can they remember or learn from their past exposure to stress? And with vermetids, there are some corals, these coral colonies that have vermetids on them. So when vermetids are present, and some coral colonies where vermetids are absent. So they have different histories, those with a history of vermetids and those without a history with vermetids. And so when I ask if corals can learn, we would expect if corals can learn, then if they've been exposed to vermetids, if they've had a history of vermetid contact, then they wouldn't react so negatively upon a second time that they were exposed to them. Whereas corals that have never seen a vermetid before, they might have a really big negative effect to that vermetid because we know vermetids negatively affect corals. So that's what this means. So if their response is dependent on history, we'd expect those corals that have no history with vermetids to show some type of large effect in different aspects or characteristics of the coral. And that would look something like this. So we would have a trait here, and a trait is something like growth rate. When these corals were exposed to vermetids, we expect for them to show a negative effect when they're in the presence of the snail. Whereas corals that have had a history of vermetids, we'd expect for them to have a small effect. So exposure to the snails don't really do anything to their growth. So whether the snails are there or if they're not there, we wouldn't see a big change in growth or any other trait. So to test how history influences the future reactions of corals, we did this thing called a reciprocal transplant study. So we took pieces of coral from these colonies without vermetids, and we took pieces of coral from colonies with vermetids. And then we transplanted them. So we took little pieces of them, we put them on a small base, and then we placed them on a colony that either did not have vermetids or did have vermetids. So for some of these, like these orange dots here, that meant that the coral was newly exposed to vermetids. And for this purple dot here, that means the corals had previously been exposed or had been in the presence of vermetids, and now they are again. And the traits, the characteristics that we looked at was coral growth in terms of skeleton. So do corals grow or shrink, or do they grow larger over the time of the experiment? Zooxanthellae densities, so are there more or fewer or less? Tissue thickness, so thinking about that layer above the skeleton, if they're thick tissues, you can think of that as like energetic reserves, like in the winter when bears get really, or in the spring, summer, whenever, when bears get really fat to prepare for a long winter, tissue thickness in this case could be thought of as a way of storing energy for any future stress, as well as microbial diversity. And in this case, because the symbiosis between corals and their microbes are tightly coupled, we're expecting changes in their microbial diversity. So the number of microbes or the composition, the communities of microbes to change would be potentially a negative effect on the coral. This is what those coral pieces looked like when they were planted on the reef. So this is an experimental unit, the experimental unit. So this is the coral that was taken from either a reef that did not have vermetids or a reef that did have vermetids, and in this case placed on a reef with vermetids. Excuse me. And you could see that with this one here. So there's a vermetid right here on one of those corals, and here's a couple of them on one of the reefs without vermetids. So the corals remain there for several months, and then we looked at the results for those different characteristics. So I'm going to go through this one slowly. So here's for looking at coral growth. So thinking about skeletal changes, if they're getting bigger or smaller. And the transplant reef. So if corals were either placed back on a reef without the snails, or if they're placed on a reef with the snails. So I'm going to show you a series of dots. These points represent the mean of the corals in the treatment. So in this case, this is the mean skeletal mass. And there's two circles here. The inner circle represents the history that the corals had with the vermetids. So in this case, the little orange circle represents that these corals had never been exposed to, have no history with vermetids. And the outer circle represents the new environment. So in this case, a reef that does not have any snails on them. So when they match, that means corals are being transplanted back onto the reef, a reef similar to the one they came from. So here, those corals that did not have a history of vermetids, we expect, again, vermetids to negatively affect these corals. And that's exactly what happens when we place corals without histories of vermetids onto a reef with vermetids. Now we're going to take corals with the history of vermetids. So the inner circle is going to be purple. And when they're transplanted onto a reef with vermetids, the outer circle is going to be purple as well. It looks like this. And then when these corals that have a history of vermetids are transplanted to reefs without vermetids, they grow a lot. And so if we put this together, we see that history or previous exposure to these snails influences their growth rate. So we see lower growth when the vermetids are present when they're transplanted to vermetid present reefs. But we also see a difference in the elevation of these lines, indicating that whether corals are originally from reefs without the snails and or if they come from reefs with the snails, that matters in terms of their growth rate. This graph means that coral skeletons grow less when they're placed with the vermetid snails, but where they come from originally matters as well for their growth. The next thing we're going to look at is microbial diversity. And one thing to keep in mind, and this is a little, maybe it's counterintuitive, maybe it's not, but increases in alpha diversity, we're going to consider a disruption of the coral microbial symbiosis. So you expect that corals and their microbes have grown in a tight symbiotic community. So when that breaks apart, that influences an influx of new bacteria to come in and take advantage of the yummy mucus that is full of sugar that microbes like to eat. So that's going to look something like this. Here we have our initial microbial community where there's 1, 2, 3, 4 bacterial species, and then an increase in diversity will have 1, 2, 3, 4, 5, 6, 7 more microbial species. And that's what this graph is showing. So when corals are placed on a reef with the vermetid snails, we see higher microbial diversity, so more bacterial species when the snails are present. And if we were to take a closer look and see who are these bacteria that are changing with the presence of the vermetids, one of them, or one of the families that are changing is this family. It's called Fusobacteriaceae. And you don't need to be able to say that, but you should know that they're ones that bacteria that are associated with no or low oxygen. And if you remember, I was telling you how the snails are kind of suffocating the corals. That's what these types of bacteria would thrive on, no oxygen environments. And we see their relative abundance. So that just means their abundance in this case. There's more of them when vermetids are present. Whereas bacteria that need oxygen, like those in the Synecoccaceae family, they're reduced. So there's fewer of those when vermetids are present. So what this is suggesting is that bacteria associated with low oxygen are increasing when corals are on these small reefs with vermetids, and other characteristics of the presence of vermetids are influencing that increase in diversity that we saw before. But if we think about both of these results together, this decrease in coral growth and this increase in bacterial diversity, what this data is suggesting is that vermetids stress coral out no matter what. So it doesn't matter the history so much, because any situation where the corals are in the presence of vermetids really negatively affect the coral. Interestingly, though, there are some traits or some characteristics that did not seem to change with transplant to the vermetid present reef. And one of those are the zooxanthellae densities, which I meant to write here. Whoops. Or that symbiodinium, the microalgae in the coral tissues, like I photographed or have the image of here, they were always higher on the corals that come from reefs without vermetids. So if they've never had a history of vermetids, they have more of the microalgae. Also, those corals that have never seen vermetids have thicker tissues. And remember, I thought of that as energetic reserves. So there's some signature of the history that a coral has experienced that's influencing their response to the microalgae in their tissues, the thickness of their tissues, and remember the elevation of those two lines, so even the coral growth rate. So why does history matter? So one question that I haven't asked and we haven't really talked about, because we've just been talking about how fantastic the variation is with corals, but we haven't really talked about, well, what makes one coral different from another coral? And one reason why that might be is because corals are genetically different. So even the same species might show some variation in their genes. Like my brother or my parents and I, even though we're related, we still look different. So I asked, do the corals have different genes? And to do that, I looked at the sequences underlying a portion of the coral. And so that's the DNA that's associated with a piece of the coral that can tell us a bit about how one coral's genes vary from another coral's genes. And the answer is yes. These corals are different even though they look identical. So this is a plot or a tree where this separation here indicates the number of base pairs. So this is a tree that's showing the difference in the genes between the corals that are listed here and the corals that are listed here. This line indicates how different they are. And what this means is that these corals are grouping together, so they have really similar genes, and these corals are grouping together, so they have really similar genes. And the length of this line indicates that this is actually kind of a big difference, to the point where they might be two different species that we didn't realize were different species because they look identical. And so this is a concept called cryptic species, and it's the idea that you can have two organisms or two things that look exactly alike, but they have different enough genes and they respond to things differently enough that they're actually different species. So if we separate the characteristics of these two different, what we think of really as species now, we have one that's resistant to colonization or resistant to the snails. So they're never found, these are the corals that don't have the vermetids on them. And then another coral species that's susceptible to the snails. So this is the coral species that does have the vermetids on them. And so what I'm hypothesizing or thinking the reason why the snails are there versus why they're not there is because of the different characteristics of the coral. The resistant corals have more of the microalgae in their tissues, they have thicker tissues, and they might calcify less. But what happens when you have thick tissues and a lot of the zooxanthellae or the microalgae, you have a lot of resources. And so if you have a lot of resources, you can come back or you can recover quickly from stressful events. And a stressful event could be the removing of some of the tissue on a coral surface. Something like this, where a coral still has some live tissue left, but then there's a partial mortality event, so part of the coral dies. But the remaining tissue, because it's thicker, because there's those high densities of algae in them, they can quickly regrow that area. So they can go from dead coral back to live coral pretty quickly. Whereas corals that are susceptible to the colonization or to the presence of vermetids, the worm snail, they don't have as many resources. Their zooxanthellae densities are lower. They have thinner tissues. Even if they calcify more, they might be more easily susceptible to damage events. So where some of that coral tissue gets scraped off, and that allows the snails to attach onto the reef. Because remember earlier I said corals can only live on the parts of the colony that have where some of the coral has died. Because the coral can't quickly regrow, that'll also mean other animals or other organisms like seaweeds can grow on that dead part of the reef. And all of this would increase the bacterial communities, so decreasing the tightly coupled relationship between corals and their microbes. And so what I'm describing to you right now is a trajectory, so the future of these small reefs. But what if we were to scale this up instead of a single colony? What would that do to the reef at large? So the entire ecosystem. So the corals, if we have lots of corals that are resistant to vermetids, so the ones that don't have a history of vermetids, we might expect that there would be higher coral cover because they'd be able to respond quickly to any type of damage or disturbance event because they have a lot of that energetic resources that allows them to quickly respond. Whereas the corals that have the snails on them, the susceptible to damage or to snail colonization, we expect that to lead to lower coral cover. It's more likely to be damaged and less likely to regrow. So what does this mean? Well, one, corals are really tricky. You can't even tell what species they are by looking at them, apparently, but snotty snails can tell them apart. So this is pretty cool because this means that even if we're not very good at IDing, other animals are better at identification are better at identification than we are.
So I have a question with that.
Yeah.
So how can they tell that difference though? Do you guys have that answer? Is it something they're letting off? Is it a bacteria that's you keep talking about the microbial community? Like, is it something that those corals have that others don't, that those snails know where to go to?
That's a great question. Maybe. So there's two parts of this that it could be. One is that the snail doesn't really care. It's just that the one type of coral is more susceptible to damage because it doesn't have it can't quickly regrow. So there's more opportunities for there to be open space for the vermetids to settle. That's hypothesis one, where the snail is actually not selecting the reef, and Mike Stephen here is wrong. The other hypothesis here is that the snail is selecting the reef to go to. And that would mean there is something about that coral that the snail is attracted to. So it could be a chemical that the bacteria are releasing or letting off that's attracting the snails there. It could be something that the coral animal is letting off to attract the snails. We don't really know. That's a big question, and in how animals in the water, when they're in this watery environment before they find their sweet spot on the reef, if you will, how they decide where to go is a huge question that I don't know the answer to.
Awesome. So that's science, right? Like, we're still always asking questions. So
right. I mean, you just learned about one project that has 10 more questions now. This is a bit of a reminder that depending on the corals that we put back on reefs, they can lead to different communities and potentially different types of ecosystems. And that's a big thought that you can ruminate with and look at this really beautiful coral picture, which hopefully will have some of these ecosystems left in the future.
Thank you so much for that. That talk was so amazing. So I just love that you have such a broad range of experience. So can you kind of tell the kids watching the experience that you have with the different coral reefs from coast to coast? Like, what kind of similarities and differences do you see? Because temperature has a lot to do with the way coral reefs function. So can you kind of explain what your experience has been?
Yeah, absolutely. So I don't know if I said it explicitly in the talk, but the work that I showed you in more detail, that was focused in French Polynesia. A few differences between the Caribbean and the Pacific. And one of those differences is in terms of the diversity of corals. So the Pacific is where corals began. So like the birthplace of all corals way back in the day. So in the Caribbean, that means that in general, the reefs in the Caribbean are less diverse than the reefs in the Pacific. So there are fewer species, and it makes it more pronounced when something bad happens because there are fewer species, and so there's not another species to take the place. Sometimes there's another species, but there's not always another species to take the place of a coral that's lost. The concept is called resilience, and one of these ideas is that with more diverse ecosystems, you see a higher resilience. And almost implicitly, because Caribbean reefs are less diverse, you sometimes see less resilience compared to the Pacific, where they're more diverse. So there's like different species between the two reefs as well. So in the Caribbean, we have staghorn corals and elkhorn corals, and they're in a genus called Acropora. And there's the same genus is present in the Pacific, but the two species that are in the Caribbean you don't see in the Pacific. So there's differences in species, but even when there's some similarities at a higher taxonomic level.
I just wanted to thank you so much. This was really eye-opening. First, I appreciate you so much for sharing your knowledge with us. I loved your explanation of, I'm gonna call it kind of like a pinball of where you went. Like it was all over the place, but it all led you to a place where you're doing the work that you need to do, and you're in a place where you're following that passion. And it's so important for you guys to realize that as students, that following your passion it's the number one priority. So if you would like to study a topic of your choosing, you can join us for our research experience. So you can follow this website here. With our research experience, you get to work with a mentor, exactly like Dr. Brown was telling you. So her main experience, like as a young scientist, she got to work with some amazing mentors, and look at how far she's come in such a short amount of time. And we can help you with that journey. So you can join us with our research experience. We are now accepting for spring, summer, and fall. So you can pick a time that works best for you. If you have any questions that you want to follow up with, you can email me at jen@headwaterscienceinstitute.org. And as always, we just want to give a very big thank you to our scientist, Dr. Anya Brown, for joining us today. Thank you so much for joining us, and I appreciate you sharing all your knowledge with us.
Thanks, my pleasure. See you.
Want to learn more about our Lunch with the Scientist program? Make sure to like and subscribe, and visit us online at www.headwaterscienceinstitute.org for a full list of our video library and curriculum.